video will be on the cell cycle, which includes mitosis. It's designed as a revision video rather than a teaching video. So if you've not yet read the relevant section in your textbook, I suggest that you go and do that, because I won't be going through everything in oodles of detail. I do expect that you have some sort of understanding of this when we start. So interphase is the major part of a cell's life cycle. It's the phase in which the cell spends the majority of its time. Interphase is divided into three phases, G1, S, and G2. In G1, the cell is physically getting larger. It is growing. In S, that's where DNA replication takes place. Again, I'm not going to go through the structure of chromosomes with you now. I expect that you've read your textbook on that section. If you've got queries, you can always let me know in the comments or through Google Classroom. The G2 phase is where the cell then prepares for cell division, whether that's mitosis or whether that's meiosis, depending on obviously where the cell is located. And then we move into the next major phase, which is mitosis. So we've got interphase followed by mitosis. Okay. So there you can see the G1, the S phase, the G2 phase, and you can see what a small proportion of a cell's life cycle is spent in cell division. The majority of the time, the cell is actually growing or replicating its DNA and preparing for mitosis. Very important to note that in the G1 phase, that is where organelles are duplicated. In the S phase, the DNA is duplicated. And in the G2 phase, that preparation for mitosis, the DNA is actually checked. So enzymes run through the DNA and check that the sequence is correct, that no errors have crept in, that there are no mutations. And if there are mutations, those mutations are corrected, usually. Sometimes they miss some. And then we move into mitosis. Now, mitosis is made up of two kind of subsections, karyokinesis and cytokinesis. Karyokinesis, karyo refers to the nucleus, so karyokinesis is the division of the nucleus, which is followed and overlaps with cytokinesis, the division of the cytoplasm. So those are seen as two separate events. Karyokinesis, the division of the nucleus, cytokinesis, the division of the cytoplasm. Karyokinesis is then divided into four phases: Prophase, Metaphase, Anaphase, and Telophase. For the exam, you've obviously got to be able to identify a cell from a diagram in each of those four phases and identify what the part of the diagram is that tells you that it is in that particular phase. You must also be able to draw a cell in any one of those phases and draw it with the correct number of chromosomes based on what the question is asking you. So if the question says with 4 chromosomes, you must be able to draw 4 chromosomes and follow those 4 chromosomes through this process. Okay, just definitions for you. So karyokinesis, the name given to the physical division of the nucleus and the chromosomes. Cytokinesis, the division of the cell, the cytoplasm, organelles, and the cell membrane. Okay, and mitosis is made up of those two subdivisions. We'll look now in a little bit more detail at the phases of mitosis. So we've mentioned before the PMAT sequence: Prophase, Metaphase, Anaphase, Telophase. We've seen this diagram before, mitosis being made up of karyokinesis and cytokinesis, and karyokinesis then breaking up into 4 phases: Prophase, Metaphase, Anaphase, Telophase. Notice that at school level, we don't do early, mid, and late prophase. We don't talk about pro-metaphase or pro-anaphase. We only look at the 4 kind of major divisions in that process. Okay, so prophase. The chromosomes condense. If you remember in interphase, your chromatin network is visible. It looks like that ball of wool, ball of string. You cannot see individual chromosomes. In prophase, those chromosomes condense, they thicken down, and they become visible as chromosomes made from chromatids joined with a centromere. The nuclear membrane, the nucleolus disintegrate. The centrioles separate and move to opposite poles, and as they do that, spindle fibers made from microtubules form between them, and all the other organelles disappear. Be very careful when you are labeling cells at other phases that you don't label the cell membrane as the nuclear membrane. So this is what we're looking for in a diagram. Cell membrane. The the nuclear membrane disintegrating, no other organelles present, only the cytoplasm is still there, the centrioles moving apart, and the spindle fibers busy forming. How would you identify this diagram if you were given this in a test or an exam? Well, the chromosomes are visible. The spindle fibers do not cover the entire cell, but they're not, the centrioles are not just right next to each other. And the nuclear membrane and the nucleolus are disappearing. Those are the crucial things that we're looking for. When we look at this in a plant cell, on the left we've got interphase. You can see there's only chromatin network. It looks like one big black ball. Whereas on the right we can actually see individual chromosomes. There's no nuclear membrane that we can see. There are no other organelles that we can see. And therefore this is a cell in prophase, a plant cell. All right, then we get to metaphase. Meta meaning middle. In the metaphase, chromosomes line up in single file along this imaginary region of the cell, which we call the equator, and the spindle fibers attach to the centromeres at the center of every chromosome. So what are we looking for in a diagram? We're looking for those chromosomes in single file. We're looking for your spindles, your centrioles to be on opposite sides of the cell, for the spindle to be completely formed, and for those chromosomes to be attached onto the spindle fibers. Notice there's no cell membrane, no nuclear membrane, only the cell membrane and the cytoplasm. Don't get those mixed up in the exam. This is what it looks like in a plant cell. By the by, we know it's a plant cell because you can see the plant cell wall has a rectangular shape in the dark orange. Okay, so anaphase. At anaphase, your centromere divides in two. The chromosomes divide in two, which means that the chromatids move in opposite directions to the poles. In order to get your mark in the exam, you have to mention that the chromosomes, or the chromatids rather, are moving to opposite poles. That phrase is crucial, and you must be able to state that like that. Cytokinesis begins at the sort of end of anaphase, into telophase and beyond. But you're unlikely to be given a cell in a test or an exam. Even in matric, where you have to do a comparison of mitosis and meiosis, you're unlikely to be given a diagram where cytokinesis is present in anaphase. They're going to give you a much simpler diagram to look at. So when they give you cytokinesis, it will be towards the end of telophase. So what are we looking for in a diagram? Well, the chromatids are clearly separated and moving in opposite directions. Those spindle fibers are clearly retracting. You're beginning to see space in the middle of the cell at the equator. Again, no organelles, no nuclear membrane, nothing like that. Okay. And this is what it looks like. Usually the diagram that you'll be given, those spindle fibers will have retracted significantly and the chromosomes will be close, if not completely, at the poles. That moves us then to telophase. Telophase is the last section in mitosis, in karyokinesis of mitosis. In telophase, the crucial thing that we're looking for is that the nucleus reforms, both the nuclear membrane and the nucleolus. And secondly, that the spindle fibers disappear. As I said, in the exam, you're likely to be given a diagram of telophase where cytokinesis is already in progress. And where cytokinesis occurs, we see the invagination, also called a cleavage furrow for animal cells, and the cell plate for plant cells. So this is what an animal cell looks like. You can see the cleavage furrow, the invagination. You can see that the nuclei have reformed. The nuclear membrane is there. The nucleolus is probably in process. There's no spindle fiber, but there are still centrioles. Okay, so that's very crucial. Make sure that you get your cell membrane and nuclear membrane labels the right way around. And then in a plant cell, you will see in the middle of this one, the cell plate reforming. If you look closely, you'll see that that cell plate, it starts in the middle of the cell and extends to left and right until it eventually meets up with the cell wall. You can see that although the chromosomes are visible, they're not as visible as they were before, and this is because the chromosomes are heading towards being chromatin network again. So they are still visible. If we go back, okay, there you can see the chromosomes are still visible, but the nuclear membrane has reformed, and of course there's the cell plate in the middle. And so this tells you that we are at telophase. As a matter of interest, look at the size of those cells. Certainly the bottom cell is quite small. Again, as the cell continues in its cell cycle and goes back into interphase, that small cell will increase in size until it is roughly the size of these two cells together. When it reaches that maximum size, it will then go through mitosis again. A really important step for you now is to do some comparative diagrams. So you need to be able to draw plant and animal cells at each of these stages: prophase, metaphase, anaphase, telophase, and cytokinesis. And it's useful to lay your diagrams out like this, to have the plant cell and animal cell, the similarities in the middle, and what is unique to each of them on either the left or the right.